All right. So up to this point, we've been we've talked about how different proteins affect different cellular processes, whether it's within the immune system or within different aspects of cell structure or cell physiology. We've talked or referred to some elements of genes. We've talked about gene expression. We've talked about how some genes are regulated. We're going to try and address this from a big picture subject called central dogma. It's kind of like a key concept within cell and molecular biology. And we'll be talking about this through the lens of antibody diversity, which is another concept we kind of touched on. And we'll be able to try and look at how we get antibody diversity within specific antigen cells that display antigen receptors. We have talked about how there are so many different kinds of possible antigen receptors that are lymphocytes can express, or antibodies that they can produce. Even ones that you've never encountered, we have lymphocytes that are equipped and prepared to respond to those unique antigens. And each one of those antigen receptors, we're going to have cells that express them. What's key to note is that this is on the order of millions, tens of millions. But if you think about it, does anyone know about how many genes we have in the human genome? A ballpark number? It's about 20,000. So there's not enough space in our genome to encode for each one of these antigen receptors separately. So there needs to, and having this number 20,000 is a good number throughout your time in BME to kind of have in your back pocket that there's about 20,000 genes within our genome. In case you ever encounter this question again, you'll be equipped and ready to shout it out loud. But there needs to be a way, given that we only have 20,000 genes, how can we produce or have millions of antigen receptors that can possibly be equipped to respond to all of these different kinds of antigens that we might encounter and that we've never encountered before? So we're going to talk about that. But first, I want to kind of set the stage for what the central dogma is. And this is going to help set the stage for our next unit for some things that we're going to take a deeper dive into. And so when we've talked about genes being expressed, or if you've heard the phrase a gene is turned on, what that means is that the DNA that corresponds to that gene has been transcribed into RNA. So DNA can beget RNA here. And from that RNA can encode for specific proteins. And so that RNA is translated into actual protein, into an actual polypeptide. So the processes here is transcription and translation. We have DNA that's transcribed into RNA, and we have RNA that is translated via translation into protein. Another part or another like arm off of central dogma. So like these two key things are like what make up central dogma. We can also say that our DNA is replicated to form more DNA. We talked about mitosis. We talked about how microtubules form these mTOCs that help pull apart the chromosomes. Excuse me. And so when a cell is undergoing cell division, that DNA needs to be replicated so that when that one cell becomes two, that we have the full genome in each of the subsequent cells that are produced. And so that process is called DNA replication. So we have transcription, translation, and replication. Those are going to be key. Those are going to be some of the key lectures that make up the third unit of this course that we start next week. But the key thing to note here, your key takeaway is that in central dogma, DNA begets RNA and RNA begets protein. So the way in which this works, where we produce nucleic acids, that's DNA and RNA. What ends up happening is that for both DNA and RNA, in order for them to be reproduced via DNA replication and during transcription, is that in both instances, there is a DNA template strand. That means that there's a strand of DNA that's being read and from which it's being used to produce a like corresponding strand of either DNA or RNA. So both processes require something, a DNA strand is being read that is then used to create the DNA or RNA. The enzymes that help mediate this process are called polymerases. As you can imagine, during DNA replication, that polymerase is called DNA polymerase. There's going to be a couple of polymerases that are involved here. There's two different kinds of DNA polymerases, but again, we're going to save that for the replication lecture. But the key thing to take away here is that DNA polymerases are critical and necessary for this DNA replication, and that they help duplicate, once that comes back, they help duplicate the DNA in preparation for cell division. And as you can imagine, in order to make RNA, that polymerase is called RNA polymerase. Some things in science, the nomenclature isn't easy, but here, thankfully, it's a little more straightforward. RNA polymerase is critical for transcription. That's a process where DNA begets RNA. And so both of these polymerases will read a DNA template strand, excuse me. And in the case of transcription, RNA polymerase will read the DNA template strand and produce a corresponding RNA strand. Are there any questions at this point so far? Yes. Great question. So the DNA template strand is the DNA that's already there within your nucleus. And so these polymerases will come in and they function differently, but they'll come in and they will interact with your DNA that's already there. DNA polymerase will make new DNA to replicate that. RNA polymerase will come in and read our DNA is a double helix. It'll read one of the DNA strands and produce an mRNA that kind of corresponds and is complementary to that. Yeah, so that's a great question. The question is, are there multiple DNA templates or DNA template strands? Yeah, so DNA replication is happening at multiple points across our DNA, across our genome. And so there's conceivably like multiple DNA templates happening simultaneously. But the main takeaway, the big picture is that DNA replication, you're just taking, if you imagine our DNA is packaged really tightly into your nucleus. You can envision, and they're packaged into chromosomes. We have 26 chromosomes. And so you can imagine that if you just unwound one of those chromosomes, it's just like a bundled up string of DNA. Just imagine we're just creating another copy of that DNA string. Does that help does that help clarify? Great. Any other questions? All right. So for a brief overview of DNA replication, what we have here, again, DNA is a double-stranded helix. So we have our double-stranded DNA here. DNA polymerase will come in. Our DNA is unwound here. And we have what's called an RNA primer. During replication, the way this will make more sense when we talk about replication, but there needs to be something there for this DNA polymerase to work on that new strand that's going to appear on the bottom half. So we have an RNA primer that comes in that helps prime the first DNA polymerase. So that DNA polymerase will come in, it'll see the primer, and from there, it'll go through and replicate and create a complementary second DNA strand right there. The second DNA polymerase will come in and replace that RNA primer with actual DNA. And so from there, we have a totally new strand of DNA that's been produced. Brief overview of transcription. Again, we have our DNA here. Instead of completely unraveling this double-stranded DNA, RNA polymerase instead will come in and wedge itself within the DNA right there. And what will end up happening is that RNA polymerase will go across and for lack of a better phrase, read the template strain of DNA. And as it does that, it's going to create an mRNA strand, a single-stranded mRNA strand that corresponds and is complementary to the DNA strand. Are there any questions so far about these two processes? This is a big picture, but we'll dive further into these next unit. Yes. Yeah, so we're going to talk about that. That's the directionality of transcription, and our DNA strands also have directionality, and that directionality is important. So we'll talk about that during we will talk about that during our transcription as well as our replication lecture as well. Great question. Yes. Yeah, so this goes back to the central dogma. DNA begets RNA, that's what transcription is. Translation is this mRNA, and we're going to talk more about this in this lecture, encodes for a protein. So this mRNA strain that was just produced for transcription will be used in the next process, translation, to create an actual polypeptide, an actual protein. All right, so as we and then the mRNA is produced, and it'll be used for translation to create the protein that it encodes for. So in order to help kind of clarify a few things, we're going to define a few key terms here. So we've referenced genes, we've referenced different how genes can encode for things. So an important thing to consider here is what a gene actually is. A gene is the entire nucleic acid sequence, in this case, the entire DNA sequence, bless you, that's necessary to produce a protein, a polypeptide. And so we've talked about, for example, during our sickle cell disease lecture way back, the very first lecture, which I'm sure feels like eons ago for you guys, when we talked about the specific beta chain of hemoglobin that undergoes a specific mutation. That beta chain for hemoglobin, there's one gene for beta chain hemoglobin. And so, and it produces that one single polypeptide, that beta chain. And so a gene is what encodes for a single protein. Now, there's going to be regions within that, there are going to be regions of our DNA that don't encode for a protein, but are transcribed. They are during transcription, there's RNA polymerase that comes in and produces an mRNA. Those are, if they don't encode for a specific protein, that's called non-coding RNA. And that's an entire area of research where those non-coding RNAs have super critical roles in biology and physiology. And we're going to talk about some of those roles in one specific lecture in the next unit. But a key thing to note here is that the DNA regions that are transcribed but do not encode for a protein, or they encode for non-coding RNAs. And what's key to note is that those are also going to be considered genes. They undergo transcription. Even though they don't encode for a protein, they also can be considered genes because of the non-coding RNA that they produce can be super important. Yes.
So just the DNA that are just never read, never transcribed, or is it always going to be gene with a functional output or non-coding?
We're going to get to that. So there's the question was, are there regions of the DNA that are not read, that do not encode for specific non-coding RNA or a specific protein? So we're actually going to touch on that, and I appreciate the question. There are, amidst the large expanse of DNA, there will be regions that do not encode for a protein and that do not undergo transcription, and largely by nature of having 20,000 genes. Again, keep that number in your back pocket. And also having about 3 billion base pairs in our DNA, there's going to be large segments of our DNA that are not transcribed and that do not encode for anything. Historically, the Human Genome Project helped determine all of these genes that we have. And historically, these regions they're not transcribed are historically called like junk DNA. But over time, we actually figured out and determined that these non-coding DNA regions have supercritical roles that affect transcription, secondarily, but are not transcribed themselves. Yes. Yeah, so there are going to be parts of our DNA that are not transcribed, but we're going to get to the introns and exons in a second. You're stealing my slides, so we'll get to that in a second. Yes. Yeah. So, oh, sorry, I need to update Slack. Historically, they were originally not considered genes, but over time, so again, this is another way of illustrating that this class is a class of half-truths, or a class that's still evolving or happening in real time, meaning that the role of non-coding RNAs is an active area of research. And historically, the areas of our DNA that were transcribed and produced these non-coding RNAs were traditionally not considered genes. However, as we've figured out really important roles for these non-coding RNAs, and we'll actually talk about a few of those key roles in future lectures, they are now starting to be considered genes because of how important those non-coding RNAs are. Does that help clarify? They were not genes before, but they are genes now, basically. So these non-coding RNAs can vary in size. There are really small ones called microRNAs. And so when we showed how mRNA is produced during transcription and that mRNA is used to produce a protein, these like small little microRNAs can actually come in and promote the degradation of mRNA. And so it can actually regulate expression of a gene by silencing it. And so they can target lots of different things. And so as we've figured out those roles for those, like a role for this one class of non-coding RNAs, now we're starting to address the genes that, the regions of DNA that correspond to them as genes. Does that help? Great question. I'm going to try and make sure that I stop for questions because I think this can be a really complicated lecture if we just fly through it. So I intend on stopping a lot. Yes. Sounds great to me. Okay. That's actually you actually asked a super important question. So I appreciate you asking that. And just to make sure I'm hearing your question properly, were you asking, does non-coding RNA correspond to non-coding RNA? Sorry, non-coding DNA, does that correspond to the non-coding RNA? Yes. So the answer is no. So non-coding RNA, so the non-coding DNA, that's going to be regions that do not undergo transcription. The non-coding DNA are these regions that were historically referred to as junk DNA. We don't refer to them that way anymore because we're actually realizing they serve pretty important roles in promoting transcription. You can actually regulate transcription in a few different ways. Some of them is these like old junk DNAs can actually help promote RNA polymerase to come in a little more easily. But non-coding DNA is not transcribed. Non-coding RNA, so because this is a hint, because it's RNA, that means it's been transcribed. That means it's encoding for, it can produce an RNA, but that RNA, non-coding means it does not produce a protein from there. So we used to think these non-coding RNAs weren't very important. Now we're understanding, that these non-coding RNAs are pretty important in biology. So non-coding RNAs, they undergo transcription because they're by nature being an RNA now. But the non-coding DNAs are going to be these vast regions of our DNA. It actually might help if I show, I'm going to advance to the next slide because I actually think that might help. These like regions of our DNA. So I'm going to move to the next slide because I think it's going to help answer your question as well. So in a previous question, there was a reference to introns and exons. So within our genes, the regions of our DNA that actually code for the subsequent protein that'll be produced, those are called exons. They only constitute about 1% of our actual genome. And so here we have gene 1 and gene 2. These regions labeled boxes A and B, those are exons, because the actual nucleic acids that are present there are going to be present within the mRNA that's going to emerge during transcription. And those same regions will actually encode for the amino acids that make up the actual protein that's going to emerge. Another segment of our DNA is oftentimes that's within our genes is also referred to as introns. This picture isn't entirely drawn to scale, but it's just meant to help you appreciate a few things. So introns are the regions of DNA that lie between the express segments, or the exons, that lie between the exons within a gene, but they do not encode, they do not contribute to the protein or the encoding of the protein. So here in gene 1, the black line between box A and B, that's the intron. Same thing with gene 2. The black line between A and B, that's the intron. It does not encode for, the DNA within the intron does not encode for the protein. Yes. Yes, that's the next part. So sorry, did you say non-coding RNA or DNA? So the non-coding RNA would be, in reference to an earlier question about these non-coding RNAs, one class of them I talked about was microRNAs. Gene 1 could be the microRNA gene, like one that I studied was microRNA 155. So gene 1 could be microRNA 155, the gene for that. And it undergoes transcription and produces that RNA, but it's not translated. So it stops there as a non-coding RNA. But the introns are part of within a gene that encodes for a protein. Does that help make sense? So by nature of being an intron, it is a part of a gene that encodes for a protein. So the next region of our DNA is extragenic. It lies between the genes. This is what was historically referred to as like junk DNA. It's the black lines that are outside, on the outside of, I'll actually step away. This would be extragenic. This would be extragenic. And this would be extragenic. It lies outside and between genes and constitutes a fair proportion of our actual DNA. Does that help answer your question? Yeah. Mm-hmm. Let me go ahead and like out loud rewrite that slide on the fly. I'm biased, but but I think it's fairly accurate to say this. The segments of DNA that encode for non-coding RNA, we're going to call those genes. Does that help clarify? I think the may be referred to might be unclear language. So regions of the DNA that produce non-coding RNA, we're going to call those genes. So the regions of our DNA that are not genes would be the extragenic DNA. And it's kind of in the name, extragenic, outside genes. So that's going to be the segment of our DNA that is not, that we will not refer to as genes. But if it produces a polypeptide, if it produces a protein, or if it produces a non-coding RNA, we're gonna call that a gene. Thank you for asking that question. Yes. You can go first, and then you can go. It might be the same question. Yes. Yeah. I think that might be my next. Your question is my next slide. Or your second question was my next slide. For your first question, it sounded like you were asking the difference between introns and extragenic. So again, introns lie between the exons. It's within the bounds of the gene. So like the gene one, if we said, these are the bounds, like we know how many base pairs gene one is. We'll say it's from here to here. And there's only 2 exons. What lies in between is the intron. This isn't really drawn to scale. As you can tell, like there's more introns than exons within our DNA, but it's the regions between the exons. Those are our introns, here and here. And it's different than extragenic because it's within the bounds of this area of our DNA, that's this gene. Yes. That is an excellent question. There are, it can vary by protein, but typically the leaving out of an exon can either produce, I'm just going to switch to the next slide because we're basically talking about the next slide. So I'm going to answer your question and your question by going through this slide, and then I'm going to look to you guys to look for confirmation. So because we have introns that actually do not encode for the protein, there needs to be a process that actually gets rid of the introns. And so that's this. And so right now, just for clarification, we're going from the DNA level now to the RNA level. So transcription here has occurred. We now have an mRNA, single-stranded mRNA strand, and we have within them 2 exons in this intron region in between. What we have here is called the primary transcript. That's what we refer to as like the naive mRNA strand that occurs right after transcription. So because again, this intron doesn't actually, this intron is not actually part of the actual protein or the functional RNA, it needs to be removed. And so this process is called splicing. Excuse me. And so what ends up happening is that there's a process by which the intron is removed and we only have exons here. And so excuse me, the process by which these introns are moved are called splicing. And what can actually happen sometimes is we can have different alternative splicing events. So if you remember, we talked about subunits and we talked about domains early on with proteins, and that subunits are usually 2 different distinct polypeptides, but domains, like if we go back to thinking about the botulinum toxin, that there's these like different domains of that enzyme. Each of those domains correspond to different exons. And so what can end up happening is that for some proteins, we can have more than 1 exon for a given domain, and some of these different exons can be removed to create different alternative splicing events and create different variants. So in this case, it's possible that during splicing, the A2 exon can be removed, producing an A1 and B variant of this protein. And a different variant could have the A1 exon removed, only having A2 and B. And so this actually produces what we call different transcript or splice variants. Does that help answer your question? Did I answer your question? All right. I'm going to stop here and give space for a couple more questions. Yes. After what? After all the introns are removed, we would call this a mature mRNA strand. At the end, that's a great question. I appreciate you asking that. At the end of splicing, we start with, on the previous slide, I'll go back 1 slide. I'm going to go back 2. This is where I meant to go, sorry. What we have here at the very top is the primary transcript right after transcription. And the indication that this is a primary transcript is that the intron is still there. At the end of splicing, what we have here is a mature mRNA transcript. And it's possible to have different of the same gene to have different splice variants that originate from the same gene, but by nature of how splicing occurs. And the way in which splicing is regulated is an entire area of research. But we do know that a single gene can sometimes produce different we sometimes call them splice variants. We might call them isoforms of this gene. Did you ever hear that? Mhm. The question is, do introns have a separate role than other than being a placeholder and then getting spliced out? I would say the right answer is probably, but to my knowledge, I don't think so. These are all great questions, by the way. All right, we're going to keep moving. And again, if a question emerges, feel free to shoot your hand in the air. So we've talked about RNA. We've referred to mRNA a few times now that emerges as a result of transcription. There's actually 3 flavors of RNA that are all really important for the process of translation, of taking RNA and producing a polypeptide, of producing protein. So the first one, again, is mRNA. mRNA stands for messenger RNA. And so it is really, as you can imagine, it's really important for translation because it's taking it is almost a direct copy of the DNA. There's a nucleotide that's unique to RNA that's not in DNA, but again, we'll get there. But it's functionally, mRNA is taking and carrying the message, it's messenger RNA, of the region of that gene that encodes for the protein. And so it's going to be basically what is, for lack of a better phrase, read during translation. And typically, each base pair, so when we're at nucleic acid, we'll typically read nucleic acids at a single base pair at a time. Base pair equals 1 nucleic acid. 3 base pairs or 3 nucleic acids equal to 1 amino acid. So when we talk about amino acids from the very first lecture, the way we get these amino acids is by reading mRNA, and we're going to read them, like you can think of it as like the frame by which the mRNA is read is by looking at 3 base pairs at a time, and those 3 base pairs are going to encode for 1 amino acid. So that amino acid that's going to be brought in is going to be brought in by transfer RNA. Transfer RNA is going to look at this, it's going to look at these 3 base pairs, and it's going to deliver and bring in the amino acid that corresponds to that codon, to those 3 base pairs. And it's going to shift over, look at these 3 base pairs, these 3 nucleic acids, and it's going to deliver the amino acid that corresponds to that. And that's how we start getting a growing polypeptide of amino acids during translation. And finally, the last flavor of RNA is ribosomal RNA. Ribosomal RNA is going to associate with proteins to form what's called a ribosome. And these ribosomes are really key machinery during translation, during the process of creating these proteins. So to demonstrate what this looks like, yes. Is it the key? What, sorry? The ribosome, a great question. So the combination of certain proteins with ribosomal RNA forms these ribosomal units. And so those ribosomes serve as these enzymes that are key for protein synthesis. So this is going to be, again, a brief overview of translation. We're going to dive deeper into this during our translation lecture. Down below, we have our mRNA. That's going to be what's produced as a result of transcription. And so right here, we have a transfer RNA that is bringing over a single amino acid. We have different transfer RNA combinations that correspond to different amino acids. And so what's going to end up happening is that we're going to have the formation of these ribosomal units that's depicted down below. And then it's going to enable this first piece of transfer RNA to come in and dock in. The next subunit of a ribosome is going to come in, and that's really going to help initiate the entire process of translation here. So once that first once that first transfer RNA is brought in, a next one comes in. Again, the little yellow circles correspond to amino acids. Those two amino acids are then going to join together to start and initiate the growing peptide, the building the string of amino acids together. And so once that second one is added on, it's just going to shift over. So it's basically just like a frame shift process where it's going to read 3 base pairs at a time on the mRNA. And each time it shifts over, another transfer RNA is going to come in that corresponds to a certain amino acid that's going to add in. And so this is largely the entire process of translation where transfer RNA is coming in, helping to read the mRNA and deliver the amino acid to produce this growing peptide. And what's also key to note is that as this peptide grows, we showed before how like proteins can like fold into these really unique structures. As this peptide is growing, it's going to start folding into its structure. So we're going to go back to this central dogma slide that we had at the beginning, and then add in a little bit of detail from what we've discussed so far. So we have that DNA is transcribed into RNA. DNA begets RNA. And we discussed how this RNA is spliced, how there's going to be the removal of introns, and how sometimes there might be different alternative splicing events that produce splice variants. Same gene, but a little bit different based on how certain exons might be spliced out. And then from there, we have RNA is translated into protein. RNA is read and begets protein. And this protein, as it's produced, is going to fold into its like functional shape, and because science, even though it's depicted here in 2D, it actually is it's a 3D process, and so these proteins will fold based on hydrophobic interactions, based on electrostatic hydrophobic interactions. And in addition to that, one thing that I didn't mention that's important to note is that these proteins will then undergo what we call post-translational modifications. Big term. All that means is that after translation, there's going to be some changes made to the protein. There might be lipids added to it. There might be sugar units that are added to it. There might be phosphorylation events that happen to it. And so like any changes to a protein right after it's been produced, after it's been translated, I'm going to use the right phrasing here, right after a protein has been translated, any changes or modifications that happen to it, we refer to as post-translational modifications. And again, not really part of such a dogma, but still important to note is that DNA is going to be replicated to make more DNA during cell division. I'm going to hit pause here and give space for a couple of questions. You guys have been asking great questions. Don't stop on me now. Yes. Yeah, so great question. So once you have your mature mRNA transcript, all that you're going to be left with is just exons all next to each other. Exactly. Yeah. No introns there. Yes. Yeah, great question. It can make either version 1 or version 2. We don't entirely know all of the mechanisms behind it, but what's important to note is that sometimes these splice variants might happen also in disease. So we talked about during Crohn's disease, NOD2 is the main receptor at play with Crohn's disease, and it's a truncated form. It's a splice variant that there's a mutation that altered transcription and subsequent translation, where now this splice variant is like super short. Instead of having a certain amount of exons, it's probably half of that. And so we don't know all of the mechanisms behind it, but we do know that it is critical for some diseases like Crohn's disease. It depends on the gene. It'll differ between genes. In some cases, you might get different splice variants. In other cases, you might consistently get one specific variant. Yes. Are you talking about like in the case of Crohn's disease? Oh, sorry. Yeah. So okay. So in that case, those exons that are spliced out, they're going to be degraded, and those RNA nucleic acids will be recycled. Yeah. Yes. Yeah. Sorry. Go ahead. So the question is, how is the amino acid on the transfer RNA? We're gonna dive deeper into that during translation, but there's a process by which, and these transfer RNAs are going to be recycled, where like if they deliver the amino acid, they're actually going to go back to a specific location and have each of those there's gonna be different flavors of transfer RNA that each correspond to an amino acid, and so that specific transfer RNA, that flavor will be identified, and the specific amino acid will be loaded onto it. But yeah, we'll dive deep into that later on, so you can save that thought. Yes. Mhm. Yeah, that is a great question. So typically genes are on the order of thousands of base pairs. How much a specific exon, how long a specific exon is, for some reason, I'm blanking off the top of my head right now how long an exon typically on average is. This is a number I know that I've known and that is just escaping me. Do either of you know on average?
Just a quick Google.
Thank you.
It says that an average vertebrate gene consists of multiple small exons separated by introns that are 10 to 100 times.
Okay, there you go. Does that help? Okay. I'm glad you asked that, because that's something I should know off the top of my head. So I'm a little frustrated that I don't have that info. Great question. The question is, can one codon encode for one amino acid, or can multiple codons encode for a specific amino acid? So typically, and we're gonna get to this later on, and we may have all, no, we didn't. Sorry, I was trying to think if we had this. There's a table that I have in my mind. I think it's during our translation lecture. What we're gonna have is we're gonna have multiple, typically, on average, multiple codons can encode for a specific amino acid. In some cases, we're going to have, during translation, there's like one specific codon, we call it the start codon, that encodes for one and only one very specific amino acid. But on average, and this is kind of a redundancy in the system where we're going to have different codons that are going to be able to encode for different amino acids. But they're all largely going to, because it's 3 base pairs, they're all going to look somewhat similar in their sequence. If that makes sense. All right, I'm going to do last question. We're going to keep going. 2 questions, sorry. Yeah, so great question. So the question is, when an exon is spliced out, is it still technically an exon? So this is only in the context where there are different splicing variants that can occur, or that can occur. Different splicing variants that can occur where we might have exons A1, A2, and B, and different variants might be A1 and B and A2 and B. They're still exons because they can encode for and contribute to the actual final protein, but there's just different variants that can occur where those exons might not be present in the final protein. That's only in some cases. That's not true for every gene or every protein. There are going to be some genes where every exon is going to be present in the final protein form. Yes. Yeah.
I was gonna ask, so when you have introns, can the genes be like overlapped to where introns would act as exons for other genes?
Can there be overlap with introns where they act as exons for other genes? what you're describing. Okay, so what you're describing is somewhat like a mutation that can occur. Sorry, we're trying to get away from saying mutation. The phrase is polymorphism, so just like change in the DNA, where there can be, whether it can be a result of a lot of different factors, but where so I mentioned like a start codon that like kind of like starts transcription. There's also going to be or translation, sorry. There also can be, there's also a stop codon that stops the process. There can be a mutation where the codon that encodes for like a specific amino acid can be, well, there can be mutation at the DNA level that encodes for an early stop codon that then leaps in and creates almost like an exon because of, if that DNA region is somehow removed during, if it's some sort of like DNA during a double-stranded DNA break, that's like a huge insult as a result of maybe like repeated radiation doses, there's different specific events that can create like a double-stranded break. During the repair mechanism, that can create potentially what you're describing, where an intron region, because of a mutation, can now encode for, can have something that it can actually be encoded for, that can alter the DNA and the subsequent protein. Does that help make sense of it?
Kind
That
of.
was a really long-winded answer. I apologize for that.
The other half of that, would you have like two start codons and then a stop codon at the end? Would that just be one gene or two different genes?
Two different genes. Whenever you have a start codon, that's typically for that one specific peptide, that one specific gene and protein.
So I could have a stop codon or a start codon and then like 100 base pairs and then another start codon and then one stop codon. Those would be considered two separate.
You would have,
genes.
within one gene, you're going to have one start codon and one stop codon. Does that
So
help?
we'd never have two different start codons.
Correct. All right, we're going to press on and I'll leave space for questions in a little bit. So, we talked about antibody diversity a little bit. This is going to be slightly, I'm going to push pause here. The entire process of antibody diversity, to me, is the most complicated process in all of immunology. I'm a little biased because like my background is innate immunology. There are going to be people in MR6 who absolutely love this. I would beg to differ. But what I want you to keep in mind is what the big picture is. The big picture of what we're about to discuss is what enables the diversity of our antigen receptors, where we only have 20,000 genes, and there's only so many genes associated with our antigen receptors. So the big picture is how do the variability in these genes enable us to have so much diversity that we can respond to tens of millions of possible pathogens. So we know based on our calculation that we only have 20,000 genes and that there's, let's say, about 80 million possible antigen receptors, that there's not one gene that encodes for one antibody or one antigen receptor. And because of alternative splicing of our mRNA, because of how little diversity that provides, this antibody diversity cannot be explained by alternative splicing. And so the question becomes like, how does our body actually do this? What's important to note is that the genes that encode for these genes are called, some of these are called immunoglobulin genes. Just think of them as our antigen receptor genes. They contain these regions of they contain genes that are super interchangeable and that have this like set library of possible genes that can be there. And so, and they're what help define the variable region and the constant region of our heavy chains and light chains. So when we talked about antibodies, we have a heavy chain and a light chain. We have constant and variable regions based on the antigen binding site versus the FC domain. Yes. The question is, how are these set of this library set of genes defined? We're going to get there. Hold that thought. what ends up happening here, and again, I'm trying to make sure that we're at the right scale constantly here. So what ends up happening is that with these set of possible genes, this library of genes that are possible here, what's going to end up happening is that the genome itself is going to be rearranged during this process, during development. That's going to give rise to slightly different antigen receptors that are going to be different between individual lymphocytes. So these possible, let's see, is it, trying to just take note of what slide I talk about that on. I'll talk about it here. The possible set of library genes is inherited through your germline DNA. So your germline DNA being what you inherit as a gamete, as the sperm and the egg come together. That's the DNA within the sperm and egg is germline DNA, and so it's going to be inherited at that level. Okay, these changes that happen during development, during the development of the fetus, those are that's called somatic mutations or somatic changes. So there's germline changes and somatic changes or somatic mutations. Germline is happening at the level of a gamete, of a sperm and egg. And you can imagine like any germline mutation, because every cell in your body originates from those germline cells, any mutation there would be present throughout your entire body. At the somatic level, like after a fetus has been is created and is growing and during development, any mutations there might be happening in specific cells or specific regions. So there's and so the library set of possible genes that we inherit is through our germline DNA. But these changes, this rearrangement that defines which possible cassettes or combinations that can make up our antigen receptors, those are somatic changes. Somatic or soma typically means body. So you can think of them as like happening when, for lack of a better phrase, we have a body, when we are a fetus, and that there's changes happening at that level, and that they differ between lymphocytes. And all of this, which is important, all of this happens in an antigen-independent way. It's happening during development, which means you're not seeing antigen. So these changes are happening in our lymphocytes in an antigen-independent way. So both heavy chain and light chain are part of the FAB domain. They're part of the antigen binding region, and they contribute, both of them contribute to antigen binding specificity. And each of them have a variable domain. So this is the FAB domain here where we have our variable regions. What's important to note is that at the actual antigen binding site or the antigen binding pocket is where we have what these regions that we call our hypervariable regions. As you can imagine, because this diversity is really important in our antigen receptors, the site where antigen binding actually happens needs to be and is hypervariable. And so it's all, the hypervariable region again is at the antigen binding site or the antigen binding pocket. So we're going to talk about this antigen receptor diversity first at the heavy chain and then at the light chain. Again, keep the big picture in mind here. We are trying to explain and understand, I'm trying to explain and help you guys understand how are we able to have so many different possibilities of antigen receptors on our lymphocytes when we only have 20,000 genes. And again, reminding you guys that the antibody diversity here that there's a whole host of possible, there's like a library of genes that can be rearranged here that then help promote the unique and specific antigen receptors that emerge. So within our heavy chain genes, there's specific genes that are associated with heavy chain, particularly with the variable region. And so they're modular, meaning that there's these possible specific DNA, specific possible combinations of DNA that are available, that are possible here. So there's going to be 3 regions of the heavy chain variable region here. We have, they're called V, D, J, and this entire process It's called VDJ recombination. We're going to stay at a pretty high level. If you're interested in trying to understand this more, or if you walk away thinking, I did not understand that, that's okay. I don't want you to feel like you have to walk away feeling like you're 100% familiar with all of this, particularly this. But this entire process is called VDJ recombination. This is one of those times where there's sometimes looking at some helpful YouTube videos, or even like looking at a picture sometimes can be really helpful. But we're going to talk about VDJ recombination here. And it's called VDJ because the genes are broken up into three parts: V, D, and J, variable segments, diversity segments, and junctional segments. I'm just going to refer to them as the V segment, D segment, and J segment. So there's 300 possible V segments, 30 possible D segments, and 6 possible J segments. What ends up happening is that at our germline DNA, we inherit these possible segments, this library of possible segments. And then at the somatic level, there's going to be rearrangement to produce one specific V segment, one specific D segment, and one specific J segment. And so it's taking our germline DNA that we inherit and then somatically doing recombination to rearrange to determine which one of these we're going to have. And it's going to be, by sheer odds, it's typically going to be different for each antigen receptor for each lymphocyte during development. What's important to note is that there's going to be specific proteins that help mediate this process. There's always going to be proteins that help with recombination, with the changing and rearrangement of DNA. During VDJ recombination is only happening in our lymphocytes. Again, because our innate immune cells don't have antigen receptors. It's going to be our T cells and B cells. And so only our lymphocytes will have are going to be able to undergo this VDJ recombination. And that for this, there's going to be proteins called RAG proteins that help mediate this recombination that happens. And so there's going to be the removal of certain segments at both the V segment, the D segment, and the J segment. And so what's going to end up happening is that we're going to have the combining of adjacent V, D, and J segments that'll then undergo transcription and RNA splicing to produce a specific V segment, a specific D segment, and a J segment with the constant domains coming in after it. Yes. Mm-hmm. Yes. Yeah. Sorry. For the recording, the question was, should we imagine in the germline DNA that there's 300 V segments, 30 D segments, and 6 J segments? The answer is yes. You should, at the germline DNA level, you should envision all of those there. But then as we are within the bone marrow, as our B cells are developing, there's going to be somatic recombination that produces the DNA that's depicted here in blue above. And then when transcription RNA splicing occurs, we're then going to produce a specific heavy chain variable region that is depicted below with V3, D4, and J2, with the constant regions coming after it. Yes.
It's only like the or are also like when you want to do to and it's false.
Yeah, that's a great question. The question was, in the animation on the slide, is the recombination events as depicted here? And the answer is yes. There's going to be specific RAG proteins that are entrusted with removing the unused V segments between V3 and D4 here. And then same with between D4 and J2. And then the ones on the outer bounds will then just get spliced out.
So, like
Yes. Yep. Yeah. I'm gonna try and move a little quicker because I think we can get through it, but I'm gonna try again. The goal is to maintain the big picture here. Light chain has only a V and J segment here, no diversity segments. So you could, in a funny way to remember this is that light chain lacks diversity. Not as funny to you guys as it was to me. I thought it was funny. But it only has V and J segments. The variable segments only have 100 possibilities. The J segments only have 4 possible ones. And so there's only 400 possible combinations here. And as we showed before, instead of calling it VDJ recombination, we would just call this VJ recombination. But it undergoes the same process by which there's going to be recombination proteins, RAG proteins that help remove and join together the VJ segments that then enable the splicing out of the unused possible segments here. So the degree of diversity here that we get these antigen receptors because of VDJ recombination. So heavy chain, we had 300 possible V, 30 possible D, and 6 possible J. So that yields about 54,000. I don't know why it says you do it. A light chain is 400 possible. And so that leaves us with about 20 million possible combinations here. That's a lot, but not quite yet there where we need to be. And so with 20 million possible, there's still enough, there's not quite enough variability that pathogens could still escape detection here. And so what's important to note, and again, this this next part that's called junctional diversity, the key part here is to maintain the big picture. So with junctional diversity, the idea is when those V and D and J segments are brought together, there is a possibility for further diversity where the junctions happen, where they come together. And so that joining together is not a perfect science. It can be imprecise. And so there can be instances where there's a double-stranded break between them, and the merging of those segments back together sometimes introduces a degree of randomness during that repair process. And so when you're putting together at least here, when you're putting together the V and the J sequence here that's depicted here in blue and orange, there's 4 possible ways that they can be brought together here. You could have the joining together in this manner, that would encode for 3 specific amino acids. It can be brought together in this manner. In this third manner, it can be brought together totally differently. That would encode for a different set of 3 amino acids. And then finally, a last possible 4th one that encodes for another set of different 3 amino acids. So again, the big picture here is that the bringing to of these segments is not perfect, and the way in which they come together can sometimes introduce more diversity that encode for different amino acids here. Finally, the rearrangement and joining of light chain is different than heavy chain. Why that is, it's not entirely clear. To the best of my knowledge, there's actually more genes associated with light chain than there are with heavy chain. And because of that, there are different mechanisms for the rearrangement of light chain that have enabled more diversity. But the key takeaway is that the rearrangement of light chain and heavy chain are different, and light chain has a little more diversity. And so what ends up happening is that there's the ability for more diversity to be introduced because of the way light chain is rearranged. And so what ends up happening is that the double-stranded breaks that happen with light chain, there's the joining together there enables for quite possibly more diversity because random nucleotides are going to be put in that try and help enable that rejoining of those segments. And so because of that, what ends up happening is that we have 4 possible, with heavy chain, we have 4 possible ways that are, going to be enable for a V and D and J to be brought together. There's going to be four possible nucleic acids that can be brought in between V and J with the light chain that then is happening on the order of 3. And so the rule of thumb here, typically to help explain the diversity beyond just VD and J recombination, is how these segments are brought together, and on the order for light chain, that there's random nucleic acids that can be brought in that help try and promote the joining of those segments. And so the rough estimate calculation is that because of this, we have now approximately 80 million possible antigen receptors because of this junctional diversity that now brings us to the functional diversity of our antigen receptors in nature. We'll see if we can do this in 5 minutes. So what we have here is that there's also typically going to be these like small hypermutations that can sometimes happen as well, where T cells and cytokines are going to stimulate B cells that are presenting a certain peptide antigen. The DNA there's within the B cell is going to be replicated as it proliferates and differentiates into a plasma cell. In the V segment, that's going to be copied. There's going to be small errors that happen during this replication process. So these small, tiny errors are going to be called somatic hypermutation. Again, somatic, soma just means body. So this is happening not in the germline, not at the gamete level, but rather after either during development or after development. In this case, this is going to be after a B cell sees antigen. And so these somatic hypermutations are going to then result in a plasma cell that might have a better affinity for the antigen. We're going to have multiple, these B cells proliferate, so there's going to be many cells. One of them might have a small mutation that might make it bind a little bit better to antigen. And so it's going to be competing with the other B cells to try and bind antigen. And this is going to be basically survival of the fittest. Which of these B cells is actually going to be binding to antigen better than the others? And so a way to illustrate this hypermutation, this competition, is we have a B cell that presents antigen to the T cell. The T cell produces cytokines that then activate this B cell, and this B cell will undergo replication and differentiation. And so what ends up happening is, as a result of this somatic hypermutation, these small errors that are introduced in the variable region, we're going to have, for example, B cells that might be a direct copy of the original B cell, but this other one that had small errors is going to be called B sub X. And so when there's a subsequent exposure, so this is also why we get boosters. When there's a subsequent exposure to antigen, B sub X might actually, those changes might actually help it bind to antigen a little bit better than the other B cells. And then that B sub X will then replicate, will present that peptide to the T cell. It'll then activated, and that will then replicate. And it's possible that there are going to be small errors that are introduced that might be B sub XY, where then maybe there might be small changes that happen that might make it bind a little bit better. And so this whole process is called affinity maturation. It's trying to promote better affinity of our antigen receptor to antigen. And what ends up happening there is there's going to be possible changes in those constant domains that were to the right of VD and J. What that is that's going to be changing what the class of antibody is. We talked about IgG, IgM, IgA, IgD, and IgE. And we talked about how B cells, the B cell receptor is going to be either IgM or IgD. The reason why is when we look at the constant domains, there's going to be a Greek letter that corresponds to the class of antibody. So mu is going to be IgM and delta is going to be IgD. This is what is going to determine whether it's IgM or IgD is going to be at the RNA level, because this is going to be determined by alternative splicing. What ends up happening is that at the RNA level, we're going to have C mu and C delta here. But then there's going to be an alternative splicing that is that's going to produce either IgM or IgD. It can happen either way. This is at the RNA level because it's alternative splicing. So it's from one B cell to the other, it could be IgM or IgD. Now, that's the class switching that happens at the RNA level. There's also class switching that happens as a result of recombination. This is at the DNA level, where after subsequent exposure to antigen, the constant domains can be recombined to go from either being IgM or IgD to now being IgG based on gamma, in rare cases IgE or IgA. This is key. The class switching that happens as a result of alternative splicing, either way could be IgM or IgD because it's just splicing that can happen either way. This is recombination. It's changes to the DNA. So once this recombination event occurs, once it switches to, say, IgG, it's going to stay that way, and that B cell will always produce IgG. And I already answered these two questions for you. Defects in VDJ recombination can lead to SCID, severe combined immunodeficiency. Another form of SCID is Omenn syndrome. Other defects in VDJ recombination can lead to different forms of cancer. Lymphoma as a general class is going to be mutated cells within our lymph system. And leukemia is going to be mutations to our immune cells that leave them in a naive precursor state. And so this VDJ recombination is super critical for the function of our lymphocytes. And these are the cases in which mutations there can happen. If this interests you, here's some folks at UVA that study this further. I went 2 minutes over time. I apologize. Thanks for sticking around, you guys. I'll stick around for any questions.